So, all right, we've made it to lecture 5, and this is the first part of Unit 2. Before we jump into lecture 5, I want to talk a little bit about Unit 2. Unit 2 is a little bit different from Unit 1 in that it has some disconnected topics. So there are several different things clumped together into Unit 2. Those would be like the fungi and the protists, diseases they cause, viruses, how viruses replicate, and then different aspects of microbial genetics. And that's it. 3 different lectures go into Unit 2. The first thing you're going to notice in Lecture 5 here today is that there will be a new kind of material that you haven't seen, and that specifically is infectious diseases and the pathogens that cause them. It might not look immediately new to you, it might not look very different, but it's different in that you have to study it differently because there are different exam questions, different quiz questions that come along with that kind of material. So I want you to learn very specific things about the pathogens and the diseases. And so before we get into those things in Lecture 5, I'll tell you more about that so you'll know what you need to know before we get into it. That's what's coming on the near horizon. Before we talk about the fungi, I just want, again, to make sure you know where we are in Unit 2. Also, I want to clarify for you what's going to be on the upcoming quiz and what's going to be on the upcoming exam. This slide aims to clarify what to study for the upcoming quiz and the exam that comes after that. So it's in 2 groups here. So what's going to be on the quiz? Well, basically the beginning of Lecture 5 is going to be on the quiz, and then again it'll be on the exam. All these other things will not be on the quiz, but they will be on the exam. And what I want you to notice in particular is part of Lecture 5 is on the quiz, and the other part is not. And so you'll see that the lecture is divided into 4 sections. The first one is about fungal biology, and then fungal diseases is the second part. Third part is protist biology, and the fourth part is protist diseases. And so the first 3 parts are up here. The fungal biology, fungal pathogens and diseases, and then protist biology. That's going to be on the quiz. But the protist pathogens and their diseases will not be on the quiz. So now we'll get into Lecture 5, and where I want to start Lecture 5, where I want to start fungal biology, is with the discussion of antimicrobial chemicals. So these are chemicals we use to usually kill or damage microorganisms. And when we think about these chemicals and the different types of chemicals out there, we can kind of start to appreciate the differences between bacteria and the fungi. I think you'll see what I mean. So let's look at antimicrobials. I want to talk a little bit about antimicrobials. In Unit 3, we will look at sort of antimicrobials of all different sorts, and we will look at the ways we control microorganisms and the ways we prevent them from growing. One thing you should know is these terms. So disinfectant, antiseptic, and antibiotic. And I will use these terms a lot. In particular, I want you to look at antibiotic. An antibiotic is a molecule that is toxic to microorganisms but does not kill a human, even if it's inside the human's body. So ideally, an antibiotic would be perfectly deadly to a microorganism and perfectly safe for us. That would be the ideal antibiotic. In reality, many of them have side effects. Some of them are extremely toxic to us, and we only use them if we must. So let's look into that, and you can read the rest of this on your own time. So how do antibiotics work? Well, generally they disrupt biological processes that go on inside microorganisms. They're going to disrupt the metabolism of bacteria. They're going to disrupt the cell walls of fungi. They're going to disrupt the cell walls of bacteria. You'll recall that your own cells do not have cell walls. So something we do that very specifically disrupts bacterial cell walls will not have the same effect on our own cells. And that allows us to have molecules that are specifically toxic to bacteria, and in some cases, fungi and protists. So antibiotics are going to be toxic to at least one microorganism, and we'll see a lot more about that. So ideally, they disrupt things like peptidoglycan. Well, every bacterium needs peptidoglycan, almost every bacterium, but humans don't. So the vast majority of prescribed antibiotics target peptidoglycan. The penicillins and the cephalosporins, and that's a huge group, and vancomycin, they all target peptidoglycan. But also, bacteria have slightly different versions of things like ribosomes and RNA polymerase and DNA polymerase. They do the same broad things that our own versions do, but since they diverged from our common ancestor, since our common ancestor lived so long ago, billions of years ago, with a B as in Bravo, we have changed a lot since then. And bacterial ribosomes are different from our ribosomes. Bacterial RNA polymerase is different from our RNA polymerase. And that allows us to make some antibiotics that specifically inhibit bacterial ribosomes without disrupting ours. So that's what we're always looking to do with antibiotics. So if we look at our phylogenetic tree, and we're going to look at this a few times today, we are over here, bacteria are over here, and the branch that separates us is here. And so all of us, we are at the end of this very long lineage, a long way away from that universal ancestor. And the bacteria are all a long way from that universal ancestor. That means that, like I've been saying, we have a lot of differences with them, and we exploit those when we make antibiotics. Yeah, if you were comparing animals to any bacteria in this tree, you would have to trace the length from here and then the length all the way to here. That would give you actual ways of measuring how different they are. Well, what about the fungi or the protists? These are the topics of Lecture 5, and they're all over here on the eukaryote side. So to get from us to any one of them, it's a much shorter path evolutionarily. We are much more similar to them. Their cells are very similar to our own cells, so there aren't as many antibiotic targets that we would have for protists and fungi. So we don't see the enormous proliferation of antifungals and antiprotist medications. We definitely do have these medications. We have found some differences we can exploit, but it is a lot harder because they are similar to us. Okay, so with that said, that is how I want you to start thinking about the fungi. They are close to us. They are so close to us, and yet they are different from us. If you look at that, if you look back at that phylogenetic tree, as I pointed out before, the fungi and the animals and the plants are all kind of grouped together. Closely grouped if you compare us to the bacteria. And what this slide has is a table that shows biochemical differences between fungi, plants, and animals. And what I want you to get out of this table is not, I don't want you to memorize it. You don't even have to read the whole table. But for the three things with arrows, these are important, distinct characteristics that we share with the fungi. And more than that, they're just things I want you to understand. I want you to know what they are. So what is a cytochrome? Where have you heard of cytochrome before? This is one of the things we have that's similar with fungi, but not with animals. Absorptive nutrition. Well, what does that mean? Well, it means taking molecules from outside and bringing them in, like what our cells have to do to get glucose and lipids and fats. Compare that to plants. They get their nutrition through photosynthesis, so they don't have to take anything from their environment that's solid or liquid. They get all their carbon from air. And so that is kind of different. They don't have to use like vesicles and all that stuff to get nutrition. And what you'll see with the fungi is that a lot of them are able to do things like break down wood and get nutrition from it. And they're very good at that. And then you should know what lysosomes are if you go back to your cell biology. So what is a fungus? Well, the quickest way to define it is to show it on a phylogenetic tree and to show the groups of organisms that all fall into that group. And it is one group. All the fungi have one common ancestor, and there's nothing else but the fungi in that group. All of them are chemoorganoheterotrophs. They have the same cellular structures we have, but they have chitinous cell walls. So cell walls made of the polymer chitin. They tend to be single-celled, and they grow in a more complicated way than what we see with bacteria. Often they have multiple growth stages, and they will look different during those stages. Some of them can reproduce sexually, where they have to find the opposite sex in order to reproduce. And that's the thing. We won't look a lot at the macroscopic fungi, but I do want you to understand what they are, and so I will show you some of that in another couple slides. What you see when you look at a mushroom is a structure that the fungi use to reproduce, and it's a collection of cells that are building this structure together, but it isn't really an organism by itself. Each individual mushroom is kind of a team effort by a lot of individual cells to try to make that structure. It's fundamentally different from like you. You are one organism. All of your cells have the same genome with tiny little differences. In a mushroom, that's not the case. The cells don't all have the same genome. They aren't all from the same one egg. So there's that. And puffballs have the same general idea. What I'm going to end with for this video is molds and yeasts, because these are common words you're going to hear, and you need to know what they are. Yeasts are single-celled, solitary fungi that act like bacteria. If you have them on a petri dish, they look like bacteria. They form little colonies. And they do, again, grow by themselves. Molds are different in that they make filamentous hyphae, and I'll show you what those are when we come back in the next video. Molds are more complex. The hyphae they make form structures called mycelia. I'll show you those. and they can also generate fungal spores, which are kind of a dormant structure that is not nearly as durable as a bacterial endospore. The key point with molds and yeasts, of course, you have to understand what they are, and I'll show you that in some detail. But the key point is that they are ways fungi can grow, but they're not separate groups. So lots of different species can grow as either yeasts or molds. So it isn't like there's the group of molds on one side and the yeasts on the other. Mold or yeast is just a form that they can take when conditions are appropriate. One example of this is Candida albicans, which we'll come to in a little bit. It's a fungus that typically grows in us and on us without causing damage. And in those cases, it typically is growing as a yeast. But when it invades tissues, for example, someone who has AIDS, who doesn't have the immune system to fight it off, if it invades the tissues, it tends to grow as a mold. And so we call that a dimorphic fungus. Okay, so we'll see all of that again later in these slides. So welcome to Unit 2, welcome to Lecture 5. We'll talk more about fungi in the next video.